Then coming to the methods of CRISPR knockout, as I already told you before, is there. That is, if the repair process is taking place by NHEJ repair process. That is, a organism, they have two repair process, NHEJ and HTR. And if the repair is taking place in NHEJ process, that is non-homology exit joining process, non-homology end joining process, if it is occurring, then what happens is we can frame shift. The gene will undergo frame shift mutation and the gene will become non-functional. So CRISPR knockouts can be developed. The advantage of CRISPR knockout, what is the advantage of knockouts? Can anyone say? What is the advantage of knockout? You can say what you have an idea. The advantage is that knockouts means they doesn't have the functional gene. So we can learn about the particular disease which is occurring as a result of the dysfunction of that particular gene. Due to the dysfunction of that particular gene, what disease has been arised, that can be studied by using knockout. Then knock-in is there, where the insertion of gene take place. And what happens when the particular gene is inserted in particular segment? What changes will occur in the organism? That can be studied by using knock-in. HDR repair process is used. Then another method is screens are used. A large number of CRISPR screens are there. These are there to find out the targets, to find out drug targets, like that. So many screens are there. Then next is CRISPR activation and interference. A and I is there. This both CRISPR activation and CRISPR interference, this is related to the upregulation and downregulation of gene. This CRISPR A, it always upregulates the gene. So what happens as a result of upregulation of this particular gene can be studied by using CRISPR A. Then CRISPR I can be used to study what happens is if the downregulation of a particular gene occurs, what happens to this human or what happens to this living organism, that can be studied by using CRISPR I. So these are the different methods that is there in the CRISPR. So coming to our main topic, that future of CRISPR is its clinical application itself. It has been studied for cancer treatment, then infectious disease, especially for COVID-19, then HIV, HPV, blood disorders, neuroscience, periodontic disease, then rare protein folding disease such as amyloidosis, that is amyloid cardiomyopathy and all that we can study. Then pre-implantation genetic diagnosis, and also for certain mitochondrial disorders. For all these purposes, CRISPR has been explored. Studies have been going on, but the correct results have yet to publish. The studies regarding their safety, long-term use is going on. So in cancer treatment, if you are saying the CRISPR Cas9, it is used to manipulate the genome, the tumor. The tumor research, it was used to manipulate the genome and to find out the tumor initiation, progression, and also metastatic phase. This CRISPR-based therapy is currently available. It is in clinical trial. It is using. Clinical trials are going on to treat leukemia, then lymphoma, and also in China, one clinical trial is completed for lung cancer. Then in United States also, by using CRISPR-based cancer immunotherapies, one clinical trial has been completed, but the results are yet to publish. The scientific world is eagerly waiting for the results to come. Then in Chinese human trial, Sichuan University's West China Hospital also, one PD-1 modified T cells is introduced to 12 patients with non-small cell lung cancer. We know that PD-1 is programmed cell death protein. So that modified, which is present in the T cell, surface of T cells. So it is modified and it is used for 12 patients to treat this non-small cell lung cancer. Then one completed study was there, but the results was to evaluate or to publish. It was the safety evaluation of PD-1 knockout. and that is PD1 knockout engineered cells that has been created, and it is used for esophageal cancer. So this is what happens in T cells, CRISPR edited T cells. This is what happening. The first is the remove blood from the patient, so as we get the T cells, the lymphocytes T cells has been collected. Then CRISPR edited T cells in the lab, that is a gene NYESO1. You maybe have an idea. It is actually New York esophageal squamous cell carcinoma 1 gene. Actually, it is a receptor. So that gene for the particular protein is inserted, and what happens is deletion also has been done. So CRISPR, by using CRISPR, modifications are done. Then CRISPR edited T cells have been, by using suitable vector, have been transfected to the host organism. And what happens is CRISPR edited T cells, they bind to cancer cell and they kill the cancer cell. That is what happens in the cancer technology or in the cancer research with CRISPR technology. In infectious disease, as we know that one of the important problem of today is, as a result of continuous use of antibiotics, we know that multiple drug resistance are there. Microbial resistance also has been there for the bacteria. So it is a very important challenge or a growing challenge. This threat can be addressed by CRISPR-9. That is, by targeting the genome of resistant strains of bacteria in a specific manner, we can avoid multi-drug resistant bacteria. And next is COVID-19. We know the pandemic, and in COVID-19 for diagnosis and also for destruction of the virus, it has been CRISPR has been used in COVID-19 because of this CRISPR specificity as well as sensitivity. Because one more point I have to say that you may be thinking, what is the advantage of this gene editing technology when compared to other, which I said in first generation and second generation, zinc finger nucleases? What is the advantage over that? The advantage is this only, that is, in the case of first generation and second generation gene editing process, we need a endonuclease specific for each strand which we are desired to cut or which we are desired to edit, corresponding to each strand of genes which we are ready to modulate. What is needed? An endonuclease is needed. But in CRISPR, that is not needed. In CRISPR, only the enzyme, already the enzyme is there, Cas9 enzyme is there. Only a guided RNA is needed. With the help of guided RNA, we can cut or we can target multiple sequences in a particular genome. That is possible. So large scale evaluation of compounds is possible. I mean, genes is possible by CRISPR Cas9 technology. So coming to our COVID-19 diagnosis, CRISPR technology, due to its high specificity and sensitivity, has been taken as a point of care interest. First FDA approved May 2020. The CRISPR Cas tool was approved by FDA. You may be familiar, to diagnose SARS-CoV-2, and that was Sherlock with 100% sensitivity and specificity. In European Union University Association's clinical evaluation, it is 100% sensitive. Another two methods was there, which is known as Dictrum in August 2020 and Detector Boost in January 2022. These are the other two diagnoses using CRISPR technology for COVID-19 that also were introduced. Then the PD1 and angiotensin converting enzyme 2 genes knockout. That knockout CRISPR-Cas technique is done, and knockouts are produced to provide what long-term immunity against COVID-19. And this engineered lymphocytes, they have been ex vivo, expanded ex vivo and also infused back to patients. And it was found out that in a study in Kuflischer University in 2021, it was tested in a trial. This PD1 ACE2 knockout was tested in a trial, and the result is yet to be published. It is not published. Then this is what happens in this COVID-19 case. The SARS-CoV genome is there, the particular protein segments are there. What happens is that our CRISPR genes, our Cas genes and CRISPR array, um, it will detect the PAM and what happens, cRNA, as well as our TR tracer RNA is there. Both helps to cut the DNA at the particular PAM region. It is cut and the virus destruction will occur. So invaded virus destruction and thereby prognosis of infection can be reduced. This can be applicable not only to COVID-19, for all disorders we can, all infectious disease we can apply this, because by using this CRISPR Cas9 technology, we can just break the DNA of the particular viral or infection causing organism. Thereby we can destruct that. So CRISPR in diagnosis of COVID-19, that is actually a method known as collateral cleavage is there. That is cleavage of the particular disease causing organism that occurs. That is the basis for diagnosis of COVID-19 by using CRISPR. The nasal swab was taken from the infected person and the particular RNA is separated and amplification is done. DNA, by using reverse transcription process, DNA is secreted. Then the presence of Cas12 as well as Cas13 is detected. Then, as I said, collateral cleavage, one process has been taken place to cleave the amount of microbes that is, or if any viral particle is present, that viral particle, and it is visualized by using either fluorescence method or lateral flow method. In therapy, as we know, what happens is the RNA degradation, then inhibition of viral gene expression occurred. Then coming to functional study by using CRISPR in COVID-19, different mutations can be detected. The SARS-CoV different mutation. We know that now Omicron, after that so many mutant variants have came for SARS-CoV, and that all variants and their effect can be detected by using CRISPR Cas9 technology. The infection can be detected. Then the role of this ACE2 HMGB1 receptors can be analyzed in wild mouse as well as in our recombinant or transgenic ACE2 mice. We can analyze. Next one is HIV. HIV, an interesting study is there actually. The first study was in HIV, and the interesting study, what I will say is that in the embryo, that is the main problem of children that have been born with AIDS is that they are getting it from their parents, no? So genetically engineered embryo has been made by a scientist in China, and the result is also published. The scientist, that is the uh, children are HIV resistant only. That was the result, but that study was not accepted because of the reason so many ethical issues arised. So many ethical issues arised, and one thing which noticed is among that twins, it was twins, among that twins, one of that death also occurred in younger age. So that becomes a problem, a debate, how to use this CRISPR technology for germ cells therapy. It arises an ethical concern. So much concern was there, so many problem arose, and um, illegal, it was considered as illegal. So many problems arised. So here HIV, CCR5, we know that chemoreceptor, chemokine receptor 5, it null blood, it is resistant to HIV entry. So we can, scientists have developed CCR5 knocking out CRISPR-Cas as an alternative to clear HIV virus. Then 2017, a trial was conducted with a non-viral delivery, and it was successfully employed to induce indels. Indels means insertion or deletion mutation in the genome from this progenitor cell, stem cells. Then surprisingly, the world's first genome edited babies, that was twin girls. They were born in 2018, and the study was done in South University of Science and Technology of China in Shenzhen. They impregnate a woman with genome edited embryos in which the pathway that HIV causes to infect has been genetically disabled. So they just impregnated the women, and as a result, they form an embryo which is HIV resistant. But the thing is that this study has China itself is saying that it is illegal. So ethical concern, due to ethical concern, this study what has uh, became stopped, or we can say the further action is doesn't take place. Then CRISPR Cas9 technology, it is used to destroy CCR5 gene in human germline we can use. So this is what is in HIV pro virus. The target sequence has been cleaved, and we know that by NHEJ process or by using HDR process, what happens is the particular inactive HN was created, and the virus which is inactive is formed, thereby controlling the infection. Then in papilloma virus also studies are there by using HPV16C6 by using CRISPR 2014 studies are there. So many studies have been conducted and going on, but the results are not came yet because we all know that CRISPR is in the growing stage only. The technology is in the growing stage only now. But we can say that the studies are going on in different for different infectious disease. uh, by this technology, what happens, we can uh, reduce the infection of the reduce the infection to a greater extent. Then for blood disorders, the HBB gene, that is hemophilia subunit beta, that gene mutation is there, that can be, that mutation is there in case of sickle cell anemia and beta thalassemia. So that can be edited by using CRISPR technology. And one approach is to focus the mutation, and other approach is to activate the GABA globulin. Thus two approach are there for treating this thalassemia as well as sickle cell anemia condition. And in phase 1 clinical trial, CD3A4 human hematopoietic cells and progenitor cells, they were gene corrected by using CRISPR, and they are administered to the patients. And another phase 1 by 2 study was conducted in which gene CD34 genes has been corrected, and it is used for severe SCD, that is sickle cell anemia. Then next is the important thing, Casgevy. Casgevy, we all know about that anyway, because it is the first FDA approved therapy that utilizes CRISPR-Cas9, the genome editing technique. Patients' hematopoietic stem cells, what is done is they are modified by using CRISPR-Cas9 technology, and it is used for sickle cell anemia. FDA approved that technology. In neuroscience also, so many research is going on, that is in DMT gene, that is dystrophin gene, which is there in muscular dystrophy condition. Studies have been conducted to find out the dystrophin, to modulate or to edit the dystrophin expression. Then studies also has been conducted to modulate SOD1 gene expression, thereby using CRISPR technology, thereby this lateral sclerosis can be controlled. Then another is SMN2. SMN2 is actually survival motor neurons, that is present in case, expressed in case of muscular atrophy and all. So by editing that particular gene which codes for this protein, it's possible to treat the particular disorder. And also for Huntington's disease also studies are going on. Then another studies which are going is for ApoE4 is there, that is apolipoprotein is there for our Alzheimer's disease. Studies are going on by modulating both amyloid precursor protein and ApoE4. Studies are going on to find out whether there is possible to cure, or there can be a solution to this Alzheimer's condition. Then periodontal disorders and also rare protein folding disorders. CRISPR has been used for targeting this oral pathogens and biofilms. And also this CRISPR technology has been used for ATTR, that is amyloid cardiac amyloidosis, which is amyloid cardiac myopathy disorder. So for that rare disorder also, studies have been conducted for using CRISPR technology, by using CRISPR technology. Then pre-implantation, as I told you before, always an ethical problem. So pre-implantation genetic disorders is a selective method to finding the embryos and to develop the embryos without any genetic abnormalities. And CRISPR has a wide application in it, but the thing is so much ethical concern of the process to go further. Then this is the potential application in treatment of medical science. If we are taking CRISPR's potential application, as I said, brain and neurological disorder, this genome editing, it aids a novel coding and non-coding translational modulators for glioblastoma multiformes, and in CVS disorders, by the by editing the mutation of calmodulin, it will helps to get rid of cardiac events. In endothelial growth factor receptor mutant lung cancer, this studies have been done. And in type 2 diabetes also studies has been done to reveal what changes occurred when this ABC8 or XIS2 expression changes, what things happen. Then in rheumatoid arthritis, viral infection, blood disease, as you know, sickle cell anemia as well as beta thalassemia. Then in liver disease, hepatitis B and C also studies are conducting. Ocular disease, studies conducted for blindness, for genetical abnormality, one brain blindness is there, disorder is there. For treating that also studies have been conducted. So what are the challenges? It is a method, and the future is what great. If we are saying the future is great, but the thing is that it has certain challenges, and the first one is immunogenicity. That is, the vector that is used can cause immunogenic problem. For that purpose, we can use non-viral vector, then that problem can be avoided, right? But the thing is that severe combined immunodeficiency syndrome, you may be knowing SCID. For treating that 18 patients, what is done is um, the scientists, they just used a viral vector. They just, sorry, that is not for severe combined immunodeficiency. In SCID, what happens is that SCID, what happens, scientists, they, it is an example of off-targeting. Scientists, they use particular this CRISPR technology they use. Then what happens is that for CID treatment, particular protein sequence has been edited, and it was found out that along with that sequence, the nearby sequence also mutation occurred. So off targeting occurred. Of what is off targeting? I am saying that is the region which other than we targeted, that region mutation occurred. And what happens is that 18 patients with SCID, 17 patients cured, very good, and the side effects was not there. But for one patient, leukemia arose as a result of off-target mutation. So off-targeting is one of the challenge of this our CRISPR therapy. And next I said immunogenicity. Immunogenicity also, it was found out that for one disorder named one enzyme deficiency disorder. I have read it. For that purpose, the scientists have been used this CRISPR Cas9 technology, and it was found out that the patient showed, they used viral vector, the patient showed severe allergic reaction, and the case was fatal. So immunogenicity is another problem. Off targeting is problem. Then unwanted mutations can occur, that is a problem. Then delivery techniques, Even if we are using non-viral vectors also, there are so many things which we need to take into account. If we are creating a liposomes also, evaluation, as we all know, it took so much time. So many evaluation parameters are there. Everything has to be correct. And the important challenge is ethics itself. Ethics is against for creating germline or genetic engineering modified embryos. So these are the difficulties that CRISPR-Cas9 technology is facing. The future is, as I told you, the future is immense for different diseases, for various disorders. CRISPR can cure the disorders, but the thing is that there is technological as well as ethical hurdles are there. That has to be that has to be properly corrected. Then only we can move forward. Then we can, in agriculture also, CRISPR technology can be used to produce very good crops, and animals, animal models, new animal knockout models or animal models can be created. Then programmable RNA targeting is possible. Then our target RNA drug can be screened. Then pathogen gene disruption, viral gene disruptions, etc., can be analyzed. Then more distant future, we can harvest pig organs for transplant patients. Then widely it may be, this CRISPR technology may be accessible to the treatment of disease. And what is happening in our near future is FDA approval for sickle cell anemia already we got. Then more CRISPR edited crops there for sale. Then approval of more clinical trials. Then the nutritional value of foods can be increased. Then the plants as well as engineered plants as well as animals can be created. Then disease resistant high yielding crops can be created, can be formed, can be developed. Then in the number of CRISPR based treatments also, the later stages of clinical trials, they are moving. A large number of trials have been conducted and it is in the final stage now. This for neurodegenerative, for cardiovascular, for diabetes, for different purposes. So its role is wide. In future, its scope is so wide. The thing is that certain issues are there. If we need to manage that issues in a correct way, in a proper way, then the future of CRISPR technology is immense. So the key takeaways from the webinar is, you may get an idea to what is CRISPR, then where do this CRISPR comes from. It is actually an immune system which is present in the bacteria. And what is the mechanism, how it works, and what are the applications then in different disorders, then what are the challenges that we are facing, and what is the future of CRISPR technology.